Well, good evening, everyone, and welcome to July's edition of our UCLA Cannabis Research Initiative Cannabis Conversations. My name is Dr. Timothy Fong, and along with Chris Evans, I am part of the steering committee of the UCLA Cannabis Research Initiative. It's terrific to be here on a summer edition. And for those of you who may have caught snippets of that entry music, that is not the UCLA fight song. That is the Italian national anthem, and there's a special reason why we have that here tonight for tonight's Cannabis Conversations. With that, let me just do a quick round of introductions. We are so excited, so pleased to invite not one, but two amazing professors tonight, along with Dr. Ziva Cooper, to talk about the brain's own cannabis. First, we have our own Christopher Evans from the UK, Stefan Hattos, professor of pharmacology here at our own UCLA and member of our steering committee, UCLA Cannabis Research Initiative. But secondly, we have Daniele Piomelli from our friends and colleagues down at UC Irvine. He is a distinguished professor of anatomy and neurobiology, the director of the Center for the Study of Cannabis at UCI. But before we get on with tonight's show, and before I turn it over to Dr. Cooper, let me have a just quick summary of a few really exciting events happening in summer 2021. Our UCLA Cannabis Research Initiative family is growing. It's growing. And in fact, we have two brand new postdoctoral fellows that we welcome in, Dr. Stephanie Lake and Dr. Connor Murray. You will be hearing a lot from these two stars in the field of cannabis research, and look for them in their work over the next year and 2 years that they are with us. Secondly, we have updates to our website, our cannabis.semo.ucla.edu. We have our cannabis grand rounds. We have our symposium from the spring, all there on the website. So please take a look at those videos. If you are starved for cannabis content, absolutely we have it there. Our Twitter handle, we're growing. We don't have a blue check yet, but we're now up to almost 426 followers. Again, our target is the 1 million follower mark. So we have a ways to go on that. Also new this summer, we have a page on Facebook. So like us there. So you'll see some updates from the lab, from the group, photos and news items on the world of cannabis. Brand new just this month. This is brand new. We are now actively recruiting humans to the lab, to the newly minted lab. On our Twitter website, we actually have the QR code and the recruitment flyer. If you are a human over the age of 21, and you have smoked cannabis in your lifetime, you may be eligible to participate in some of our NIH-funded studies taking place inside the UCLA Cannabis Research Initiative laboratories. So take a look at that and please call. And that's that phone number, the 310-794-1563 phone number for more information on that. Lastly, upcoming events, we do have websites for new things. We will have a fall grand rounds. And we will have more cannabis conversations throughout the rest of the year. If there is a specific topic, something you want to know more about, something you want to learn about specific to the world of cannabis, please contact us on our Twitter page, on our Facebook, or drop us an email. We're absolutely always looking for more topics that you, the audience, want to learn more. But enough of that. Let us turn this over now to Dr. Ziva Cooper, our MC tonight and host, bring on the cannabis conversations about the brain's own cannabis. Thank you. Good night. And I turn it over to you, Dr. Cooper.
Dr. Fong, thank you so much for a wonderful introduction. We are so excited to have you join our series, Cannabis Conversations. Unfortunately, it's still during COVID, highlighting the most pressing questions related to cannabis. Today, we'll be talking with Dr. Daniele Piomelli about his decades work discovering essential elements of the endocannabinoid system, which we'll learn more about, and why it's really critical to how our brains and bodies work. He is the director of the UC Irvine Center called Investigating Cannabinoids Across the Lifespan, which we will learn more about today. And by the end of today's conversation, we'll have learned about what is the brain's cannabis, why it's important for health and functioning, how it interacts with nature's cannabis, and how it can be leveraged to develop medicines. For the conversation, we invite you to click on the tiles in the upper right corner of your screen to turn on the gallery view so you can see all three of us at all times. Also, feel free to add questions in the Q&A box. You might notice that I'm keeping an eye on this box during the conversation, and we've already received many questions, in fact, over 30 questions, and we'll try our best to respond to everyone's comments and questions over the course of the hour. We are also really grateful to you, our audience members, for attending these events and for your support. You will notice a link in the chat box that can be used throughout the session to help support the work that we do at the UCLA Cannabis Research Initiative. And this support, of course, helps us to continue to have these types of events. So, Dr. Piomelli, welcome. It's so good to see you. We're excited to have this conversation with you focused on the brain's own cannabis.
Thank you for having me.
So as we get into this topic, let's first talk about your choice of title, the brain's own cannabis. Can you tell us a little bit about the meaning behind the title of the conversation today?
When we think about cannabis, we think, of course, about the plant and the chemical constituents of the plant and what makes the plant so unique in many ways. People use it for different purposes. And sometimes people who use it don't realize that the reason why cannabis works the way it works is because we have a mirror to cannabis in our brain. That is, we have a system that is able to recognize the chemical constituents of cannabis. And particularly, there is one such constituent called tetrahydrocannabinol, THC. Everyone knows that. But THC works by binding to receptors, so proteins. They are sitting on the surface of cells. And these cannabinoid receptors, as they are called, are just one part of a whole signaling system that the brain uses that is based on molecules that do pretty much what THC in cannabis does. Of course, they do it in more selective ways, but that's the logic is very much the same. And one thing I would like to add is that, yeah, I said the brain, but I misspoke because actually the endocannabinoid system, as is called, is present all over the body. So when THC interacts with our body, it calls into action pretty much every aspect of our physiology.
I noticed that you specifically touched on THC and how it interacts with our body's cannabinoid system. There is so much interest in some of these other chemicals in the cannabis plant, like CBD, cannabidiol, for instance. Does that interact with our body's cannabinoid system?
CBD interacts with the cannabinoid system, or the endogenous cannabinoid system or endocannabinoid system, but not directly. So it does probably a couple of things. It can bind to the same receptors that THC bind. It does so in a subtle way, probably inhibiting or reducing the actions of THC, but it can also do something else. It can potentiate, if you wish, or can enhance the actions of our own endocannabinoids, and one in particular called anandamide, and by blocking the degradation of this compound. And this is not a major effect of the CBD, in my opinion, but it's one that has to be considered among the very many things that CBD does.
So we actually got a question from one of our audience members about how CBD might do this. And if you know of, in your experience, have you seen studies that demonstrate that CBD is actually able to enhance these endocannabinoids, the body's own cannabinoid molecules?
Yes, of course. There are studies, and not very many, but they all go in the same direction. They show that that CBD can interact with a protein called FAAH or FA, which destroys one of the endocannabinoids. This molecule that is destroyed by FA is anandamide, the molecule I mentioned before, and is one of the two main endocannabinoids. So people have shown that if the CBD can block FA, which leads to an increase in anandamide. If you increase anandamide, you are indirectly producing effects similar to those of THC.
So interesting. Dr. Evans, so Dr. Evans, you're a opioid pharmacologist.
Yes.
And so you have a very similar experience studying the body's own opioid system. So does some of what Dr. Piomelli is talking about resonate with what you've also seen in the opioid system?
Very much so. We would probably not know about the endogenous opioid system had there not been the opium poppy. And maybe we still wouldn't know much about the cannabinoid system and its effects if there wasn't cannabis, right, Daniele? You know, it really, the plants have really led us to understand the endogenous systems. And without them, we would probably, they'd still be receptors which we wouldn't really understand so well if we hadn't had these reactions to these plants. So I think that's very important that you know these plants has really driven science. It's really driven us to understand these systems. So
yeah, it's a fascinating. I'm sorry, I didn't mean to interrupt. Please go ahead.
No, go ahead. I finished.
It's a fascinating scientific history, Chris, as you know. The morphine itself, which is the active constituent, the main active constituent of opium, was discovered back in the 19th century and was characterized painstakingly over the years. And then in the 70s, as you remember, of the last century, so now 50 years ago, people discovered that we have our own system that mimics the effects of morphine, and these are, of course, the enkephalins, the endorphins, the endomorphins, et cetera, et cetera. The story with cannabis is similar, but a little bit more, a little slower, because it took a long time to science to understand that THC, which was isolated back in 1944 the first time, to understand how THC interacts with the body. Because you see, one thing about THC that is funny is that it's actually kind of a fatty molecule. It's a fat-like molecule. So a lot of folks who were working on THC at the time thought, okay, so it's a fatty molecule. It cannot have its own receptor protein. It must be interacting with the lipid membranes, the membranes that surround the cell. They're called, the cell membranes are made by fat. And that was the thought for a long time. In fact, it was by chance that folks discovered the cannabinoid receptor, the first cannabinoid receptor, which is now called CB1. It was really a serendipitous discovery, as we say in science. It was essentially pure luck. And once the receptor was discovered, now people started to think, well, if they're just like they did for the opiates, there must be an endogenous system. We must have our own intrinsic system that activates these receptors, right? That's why probably evolution put that receptor there. And so people started to look. And again, because it was unusual for an active molecule to be a fat-like molecule, people didn't look in the right place. They were looking for peptides, just like they'd done for the opioids. Peptides are small proteins, essentially, and very sort of water-soluble proteins. And that's not what happens. The endogenous cannabinoids actually turn out to be, just like THC, fat molecules. They are produced from, the lipid membrane that surrounds our cells and act on the cannabinoid receptor, and then they are rapidly eliminated.
Well, you know, one of the theories before they discovered the opiate receptor was that opiates were just inserting them, the opiate drugs like morphine were just inserting themselves into membranes and thereby having their effects on analgesia and reward like they do. But, when they found the receptor, then they could find the peptides and that's how it went. So the sequence of events was the same with the cannabinoid system as well, right? They found the receptor first, and then you and others were finding the ligands for these receptors.
Yeah, no, that's exactly the same sequence. We are a little bit retarded in sense
Yeah.
that we were delayed.
Yeah, you were delayed.
About, I would say probably 30 years, no, maybe less, 20 years later. Yeah. But I think the reason why we were delayed is because, as I said, people were looking in the wrong place. And we scientists have that drawback. We look where there is light, and sometimes we just have to turn the light in a different place.
Well, I think the cannabinoid system is more complicated as well. it's a more difficult system to study. But you know, just because it's you're the endogenous the compound which compounds of fat are derived from fats, and with peptides it was a little easier to I think find the systems which were working. So I think we had it a little easier than you guys. So
so as the non-molecular pharmacologist here, let me just ask you guys, you're talking about the differences, the important differences between opioids that are peptides, that are proteins, and then the cannabinoids like anandamide that are fatty, that are made from fat. So are there other examples in our body of these of signaling molecules like cannabinoids that are fat? And what is the difference, or how is it functionally different, the fat versus the peptide versus the amino acids like the opioids? Is that important, the fact that they're made out of fatty material?
Chris, may I take this?
I think you should take this, and maybe I'll add on a little after. Yeah.
So the first question, do we have other signals that come from fat? Yes, we do. That's how I actually started my life in science, was looking at another lipid-derived set of lipid-derived molecules called arachidonic acid metabolites, which are products of a lipid or a fatty acid that is present in the membranes and is transformed in particular ways, and it goes out of the cell and does wonderful things all over the body. That's how I started in grad school, and that was originally my intention to stay there until the endocannabinoid system, the first endocannabinoid actually was identified back in '92. But your question, I think, is a lot more interesting. The second part of the question is a lot more interesting because beyond my own autobiographical stories, it goes to the core of what the endocannabinoid system does and how different it is, just like Chris said. So when we talk about peptides or any other classical transmitter, we are talking about molecules that are water soluble, and they are put into little vesicles that are held in the in the action place of the neuron, which is the synapse, right? That's where the action is. So these little vesicles contain these transmitters, and when electricity arrives at the end of this of the synapse, they are released just through the electrical stimulus. There are complex machinery that work there, and they're released and they activate receptors on the next cell. So that is true for basically all transmitters in the body. Molecules like dopamine or serotonin, which I'm sure all our audience must have heard about, and also the peptides. There are differences that I'm sure Chris would be able to talk about for many hours, but fundamentally the mechanism is the same. The endocannabinoids work in reverse. So what happens is that they are produced when the main transmitters have stopped their action. So they are produced, most of them, on the other side of the synapse. So they are produced when the transmitters arrive, activate their receptors, they stimulate biochemical pathways that lead to the production of these fatty molecules, and these fatty molecules do what they do, they go around. Some of them go back to where the original transmitter came from, which is called the presynaptic ending, and tell the presynaptic ending, look, guys, we have been activated, you can relax now. That's pretty much what most endocannabinoids do in this sort of signaling mode. Or they can go farther away. They can go to, say, the glial cell next door. Glial are cells that support the neurons and in a variety of important ways. Or they can go even to further away to other neurons nearby. And different endocannabinoids do these two different things. So back to your question, I think that one of the fascinating aspects about the endocannabinoids that also distinguish them from other transmitters, including the opioids, is the fact that they work in this weird way. They are really bizarre that way because they work as retrograde signals in many cases. Retrograde meaning that unlike transmitters that go from point A to point B, so an anterograde transmission, they go from point B to point A. So hence it's a retrograde signal, signaling to the presynaptic ending what is happening postsynaptically.
And I want to follow up on that really important point. Dr. Piomelli, I'm getting some notification from our audience members that right now you're speaking into the microphone really nicely. Before you were a little bit far away. So right now is perfect. So this signaling that you're talking about, where it's kind of like a backward signaling, why is that important? Or what is that? Because does that indicate that it's regulating some system, it's putting the brakes on a system, or it's telling a certain system to go? Or what? How come that's relevant or important to how our body and brains function?
It is important because it's a very subtle but fundamental modulator of what the neurons tell to one another. So one neuron tells to another neuron, act or stop acting. So it would be an excitation or an inhibition. But the brain needs more information than that. And what the endocannabinoid system does, it provides this kind of modulatory information. So it's not just act or stop acting, but act a little bit more or stop acting a little bit faster. These are all subtle aspects that we may not consider important at first, but there are a lot of subtleties in the brain. The brain is a very complicated place. So without this modulation, I think that our lives would be fundamentally different. And we know that because we know of people who have their endocannabinoid system is blocked. We can block it with drugs, and we know that they have a hard time in their daily tasks, for example. We also know that some folks in which the endocannabinoid system is enhanced, and they have also different ways of looking at the world. So even though you don't die, if you remove the endocannabinoid system, your life would be a lot different. And I think a lot worse in terms of living a fulfilling, emotionally fulfilling life.
That's an interesting point too, because we've gotten a couple of questions from audience members about this idea that some people or some disease states, there's an endocannabinoid deficiency, right? So there's this idea that people are learning more and more about in the regular community that some diseases might be marked with lower endocannabinoid levels. Is there evidence to this? And what types of diseases do we usually see this in?
Well, there is no evidence for a body-wise endocannabinoid deficiency, but because endocannabinoids can go up and down in a variety of different ways, one can have a deficiency in a specific part of the body or in a specific portion of the brain. And that particular deficiency, yes, would have, I think, substantial effects, pathological effects. What we do know is that there are folks who actually have more of an endocannabinoid system. The system is active in a greater way. And this we know because the enzyme I mentioned before, FAAH or FA, which destroys anandamide, that protein can come in a mutated form. So there is a fairly common mutation that it causes the protein to be malfunctional. So it is dysfunctional. It's not fully active. So when that happens, of course, we have more anandamide. So people have studied this mutation, and the mutation is distributed unequally in the world. But people who have it tend to be a little bit more carefree. They have less anxiety. They tend to be less worried about something aversive in the environment, and they tend to feel a little bit less pain. An extreme case is a lady in Scotland, and she's a single case for now, but she is not the same mutation. She has another mutation that is evidently very rare, unfortunately, I should say. And this lady is a really interesting story. This lady needed some surgery. I think she needed knee surgery. She was in her 70s at the time. And she went to the doctor. The doctor said, okay, sure, this is a schedule and this is your painkillers. And she looked at the doctor and said, why are you giving me this? I said, well, for your pain. I said, I don't know what pain is. I never experienced pain. She went through birth, she birthed 2 children without any analgesic. And so, scientists in Scotland started to be really excited about this. So they analyzed her genome and they found that this lady is perfectly healthy. She has one problem. She has no FA. No FA, the enzyme is gone. So she has very high levels of endocannabinoids. And so although she's only one person, she's very cheerful. She's never in a bad mood. She has this incredible lack of pain. And also she heals, her wounds heal very quickly. And that's something that is also interesting, that doesn't involve the endocannabinoid system proper, but some other lipid signals that is also destroyed by FA.
So sorry, Chris, I was going to ask if there's something similar with the opioid system.
I don't know of one. I mean, people are trying to develop drugs which enhance the endogenous opioid system, like drugs such as Daniele was talking about, which block the breakdown of the peptides, or they actually tweak the binding site for the opioids so that they bind a little more tightly. They're called allosteric modulators. That I know it's a difficult term, but allosteric means it's not in at the place where the molecule, like the opioid, is binding. It's a slightly different place in the protein, and it sort of tweaks it so it makes it bind a little better and a little longer. So they're trying to develop these kind of drugs to enhance the endogenous opioid system, but there isn't really examples of like this lady from Scotland, who has this mutation in the FA, which breaks down the cannabinoids. There isn't really any examples of that in the opiate system. So you're lucky there, Daniele. You got a leg up on the opiate system there. But I guess my question is, why do you think that when you give cannabis, you don't have the same efficacy as this woman was experiencing in analgesia?
That's a very, very important question. I think that this particular mutation is not really reflective of the role of the endocannabinoid system. There might be other factors at play. The first mutation, which is called C385A, the technical name for it. The other mutation, which is a lot more common, that one I think is closer to what happens if people use cannabis. But I think you pointed out something really, really important before. You mentioned that people have been trying to stop the destruction of the endogenous opioids with drugs. And the enzyme that does this job is called enkephalinase. It's actually several types, but the enkephalinase inhibitors were developed actually in France. I was working in France at the time. And I had the pleasure to meet the person who discovered these first enkephalinase inhibitors. I actually worked with one of them, and the pharmacologist Jean-Charles Schwartz, and with the chemist Bernard Roch, and that gave me the idea of developing inhibitors for FA, and that's how I got involved in studying the very first inhibitors for FA, which we published now many years ago. Just because of the example of the opioid system. That's, I think, the beautiful part about science is that we all are standing on someone else's shoulder, and that's it's a lovely. I think it's a beautiful social endeavor.
So Daniele, there was a disaster with one of the FA inhibitors. Do you want to relay that to the audience, because that really put a dash on the development, which I think is a really good strategy for enhancing the endogenous cannabinoid system?
Well, I don't know if I should thank you for bringing that up.
Wait, can we step back?
But anyway, so it was, at the end of the day, it's all good, because it turned out that it had nothing to do with FA. It was a company that was doing something very poorly, working with a contract research organization that did the clinical work that was very unprofessional in my opinion. I should say, actually, I was involved in this very much, because as often happens, success has many fathers, but failure only gets one. So I was the person everyone came to ask when the issue arose. They came to ask me, so what is happening? And the French did, because this happened in France as well. At the time I was here in the United States, but this clinical trial happened in France, and the Contra Research Organization who did the work, sloppy work if you ask me, they contacted me to help them out. I said, I looked at the data. I said, you guys are crazy. What are you doing? Why are you doing this way? And so to make a long story short, they messed it up big time. This compound had a bunch of, this molecule that they used had a bunch of, off-target effects, which is what the term we used to say that it's a dirty compound, essentially. It was very dirty and was poorly characterized. And the clinical trial was also very, very poor. So that ended up in tragedy. What Chris is referring to is actually the death of one person in Rennes, in the north of France, and six other people, five other people being hurt. One person made it through unscathed. I think is the one person who didn't take the drug and hid it in the plant, instead of taking it through the pill in the plant. But I think it was a catastrophe. I'm laughing now, but for a whole year I was in, and still now, I mean, people are still asking me about this, even though the Food and Drug Administration and the European equivalent called the EMA, clearly indicated that we know that phi is not involved. Still, it was a shadow that really we did not need, and it put really a big halt in the development of these molecules.
Dr. Piomelli, can we take a little step back and talk about what this molecule was? So the molecule was a drug that blocks Pha, the enzyme that degrades the body's own natural cannabinoid called anandamide.
Yeah.
And you were instrumental in developing this class of drugs, these enzymes, or these enzyme blockers to actually promote increases in people's endocannabinoid levels. And so your work in this was to look at how these drugs might be helpful for what types of indications.
Yeah, so let me just clarify for our audience what the logic behind this is. So there are different ways of making drugs, but one way that I prefer is the one where you make a molecule that does not have per se much of an effect. But what it does, it tweaks the effects of our own protective systems. So we have a bunch of systems that are homeostatic or protective, okay? And we can tweak them up if we want to. There are ways like, for example, blocking an enzyme that end up having that kind of an effect. And that was our thinking when we decided to develop the first inhibitor for this enzyme. And I didn't do this on my own. Of course, was an effort, a team, and very good friends in Italy, chemists in Italy were fundamental, instrumental to this. But the idea there was actually twofold. One, a curiosity. I wanted to use this to understand what anandamide, the very first endocannabinoid to be discovered, albeit not the only one, the anandamide, I wanted to know what anandamide does. And in order to do that, the idea was I'm going to boost it and see what happens. Because if I boost the normal action, I should be able to get some kind of sense of where we're going there, what it's doing normally. So that was the first reason, curiosity. The second reason was that, well, say if we are lucky, and actually what anandamide does is protective, which at the time we didn't know, it was 20 years ago, we didn't know that. If anandamide is indeed protective, then it could become a drug. And what we discovered when we made the first Pha inhibitor, a compound called URB597, when we made that inhibitor, we had a hard time in finding anything because it doesn't do much. It doesn't do anything that what THC does, except for a few things. And I'll explain that why that is the case. And the one thing that it does very well, it reduces anxiety. It reduces depression. It improves mood. So it's one sliver of what the endocannabinoid system does, regulation of mood, anxiety, and stress responses. And the reason why it doesn't do everything that THC does, if you think about it, is very simple. THC binds to all cannabinoid receptors. When one uses cannabis or takes cannabis as a medicine, it activates all cannabinoid receptors, doesn't have any choice, right? But when you block the enzyme, again, as I said before, you're not activating indiscriminately the receptor. You are just activating the receptor that sees anandamide where anandamide is produced. So that is the little trick that makes the difference. So now with the phi inhibitors, what we could find was that they reduce anxiety, they alleviate depression, and they have a bunch of other interesting things without having the side effects of THC, because they don't cause intoxication. They don't cause the state of, well, look, it's a state of intoxication and some people actually desire, and that's okay. It's okay with me. But if you don't want the state of intoxication and you want an antidepressant or an anti-anxiety molecule, I think one should go for something else. And I think the phi inhibitor might be that something. And I have to say that after we made that first discovery, which was back in 2002, slowly but surely there was increasing interest in both academia and industry on this. And now Johnson & Johnson has a phi inhibitor that gave a very interesting response in clinical trials, initial clinical trials. These are called phase 2 trials in social anxiety, suggesting that indeed what we discovered 20 years ago working with rats and mice could indeed be true for people. And keeping our fingers crossed, things can still go wrong, but it was a pretty bumpy drive. So after 20 years, we may have possibly a new therapeutic for anxiety, which we really need.
So a good friend of ours, Dr. Abrams, typed in the chat box, and Dr. Abrams was actually with us a couple months ago doing COVID conversations. And he said on this note, he asked, even before we started talking about these phi inhibitors, which would increase endocannabinoid levels, he said, what about just the idea of giving people anandamide? Has anybody done this? Has anybody given people endocannabinoids?
Well, it's hard to give people endocannabinoids, and that's the reason is because really these compounds are so short-lived. There is a built-in mechanism whereby they are destroyed by the body. There is all these enzymes I mentioned. Phi is one of them. Destroy them very quickly. So if you were to take a pill with anandamide, it wouldn't even get to our circulation. We would have probably to inject anandamide directly in the brain, which is not something I recommend. And besides, again, in that case, we might as well get THC, because THC does the same thing. If you want to indiscriminately activate the cannabinoid receptors, you use THC, you're better off. The problem with THC, there are several problems with THC. It's not just the indiscriminate activation of the receptor, but also the fact that it actually causes the receptor to we say desensitize, which means that basically loses sensitivity, becomes less able to produce an effect over time. And that problem, which we refer to also as tolerance, is a problem, because then you need, in order to achieve the same effect, you have to increase the doses. So now it's not always a problem. If you keep the doses of THC low, it doesn't usually affect the response. But it is a problem, especially in non-medical recreational use, and folks who use too much THC end up having, depending on THC, an increase in the dose of THC. And although that is not necessarily lethal, because THC doesn't really kill people, it's a burden on you, because you're depending on this drug.
It's exactly in line with one of the questions that we're getting, is this idea of tolerance, of needing higher doses over time when you're using something like an opioid or like THC. And if you're tweaking the body's own system, like using a phi inhibitor, do you not develop that type of tolerance as quickly or at all compared to using THC? And I don't know if this is the same, Chris, with opioids as well. If there are systems where you can tweak the metabolism or increase the body's own opioid system, natural opioids, if you wouldn't see tolerance as quickly.
Actually, I think probably Daniele is going to tell you the same, that the tolerance systems, when you tweak endogenous systems, seem to be much reduced, or if, you don't see them. You don't see that tolerance happening. And I mean, the problems with tolerance and the adaptations which occur when you take THC and high concentrations of THC is that there can be a withdrawal syndrome as well. And with opioids, that's a real problem. The withdrawal syndrome is, as most of you probably have heard, is really unpleasant for those going through opioid withdrawal. Is really unpleasant. There's shaking, the opposite effects actually of being on the drug. And that's what tolerance usually gets, because you have these adaptations which occur and you have insensitivity of the endogenous systems. So they just don't work so well, and everything's compensated. And when you stop the drug, you often have the opposite effects. Now with opioids, this withdrawal syndrome is very, very strong. With cannabinoids, it's much less strong. And I'll let Daniele talk a little bit about the withdrawal syndrome, any withdrawal syndromes, but he probably knows those a lot better than I do for the cannabinoid system. But they are much less intense than you get with opioids, but they do exist. There are some symptoms which people can get when they stop taking their THC.
Chris is absolutely right. There are withdrawal symptoms with THC as well. And actually, in this trio here, Ziva is the expert, because she used to work at Columbia University, where Meg Haney and her did some fundamental work in this, showing there are withdrawal symptoms, and like with other folks. They're subtler, they're more subtle than those induced by opioid withdrawal, but they're a problem. They're still a problem. So there is such a thing as cannabis use disorder, CUD, and that is not something that I think we should underestimate. It is problematic. What makes me worry about the CUD is the fact that it's much less, cannabis is considered rightly, I believe, a much less toxic plant than opium is, much less likely to cause immediate strong effects, and I think that is the truth. But I think people maybe underestimate the more subtle effects that it causes, and they may underestimate also the fact that so many people are using cannabis now, either medically or recreationally, that the cases of use disorder are starting to creep up. And I'm not especially concerned about it, but I still think this is something that we should keep our eyes on, especially now, because I was just reading today on the New York Times that the number of deaths for opioid overdose, particularly fentanyl overdose, this last year, possibly as a result of the COVID crisis, has skyrocketed. And so I think it's very, very important to us to understand how cannabis, which is so widely used, and fentanyl, which is unfortunately also so widely used illegally, how they interact. This interaction could be a positive interaction in the sense that cannabis may actually help, or it could be a negative interaction. And in fact, again, Ziva has done some really pioneering work on this. And Ziva, I'm going to say what you did, and I'm going to take credit for it. No, not really. I'm going to say what you did. But your work was really very important in this sense, because, to make it a long story short, she showed that actually if you combine an opioid and the cannabinoid, you get a greater pain-killing effect, but you don't necessarily get a greater sort of rewarding effect, or a greater abusing effect. So it may be good news, right, Ziva?
So it's interesting because I just looked, I did a quick peek today, Dr. Piomelli, about some things that you were mentioned of in the news. And I noticed your 2017 editorial in the LA Times, which is titled, "Can Pot Blunt the Opioid Epidemic? We don't know because the federal government blocks research." So that was 2017. We're 2021 today. Yeah. Do you think we've made any headway in this?
Hardly. I mean, honestly, and not to flatter you, but I think your work is one of the few, bright lights in an otherwise fairly gray situation. But what we do know now is epidemiological studies and economy studies have shown, which seem reasonable and seem very robust to me, although I'm not an expert in those fields, have shown that states where cannabis is both legal and available have less use of opioids and in general prescription drugs. That is a direction where we want to go, make sure that those data are either correct or incorrect. You know how science works. Sometimes we are wrong first and we correct ourselves. But the studies that I've seen, it's not just one or two, it's actually now a whole series of them going all in the same direction. States where cannabis is available, so not just legal, it has to be available to people, tend to have less prescription drug use and less opioid use. But I would not take this necessarily to mean that we are out of the woods. And okay, so cannabis now is a solution to the opioid problem because we really don't understand how these two systems interact. And I want to go back to basic science for a minute, because I think what the opioid system and the cannabinoid system do in the body is like two cousins. They really are very similar in many of the things they do, but they were separated at birth, two brothers separated at birth. So they go in their own direction. And so some of the things that they do are similar, some others are different. And we at the basic science level, we need to understand better how these systems interact, because that way I think we'll have a better sense of how opioids like fentanyl or carfentanil that are used now indiscriminately by a bunch of folks and cannabis can either interact positively or negatively.
And Chris, actually, you have spent decades interested in understanding opioid use disorder in animal models and withdrawal and dependence. So is this encouraging news for you about the potential for cannabis or the chemicals in the cannabis plant to maybe help to curtail this opioid epidemic?
Yeah, I would be more excited to see the FAR inhibitor given the genetic results, to tell you the truth, rather than THC. But I've been following the epidemiology in states which have legalized and used, and in Colorado initially there was a dampening of the overdose deaths from opioids, but now after a couple of years this has gone up. And it's now sort of exceeding other states. So it's a little worrying that the initial data was quite strong that the cannabis was going to be kind of protective, but I think the more recent data is much less certain about that from what I've seen, and I think that it's a complex story because with cannabis, there's it's not necessarily a gateway to other drugs, and that's been certainly demonstrated. It's the most available drug, but maybe there is some kind of interactions with other drugs, maybe less cognitive awareness, maybe more mistakes made, maybe it's not a clear-cut answer, I think, to have cannabis substituting for opioids. And opioids do have very, very different effects as well. They're much more potent at I think the anxiolytic effects and the anxiogenic effects than cannabinoids, but then the withdrawal effects are much worse, of course. So that makes you continue taking the drug, whereas cannabis doesn't have that sort of driver as much, that negative affect that you have when you stop the drug. But I think that cannabis should be one of the first line drugs, to tell you the truth. I've always said that. It's one of the least problematic compared with opiates, for sure. And I think there's going to be a role for it. But as I said, I think that from the data that I've seen, the FAR inhibitors would be the way to go, as opposed to cannabis as a sort of a drug.
Interesting.
Just add one thought to what Chris just said, with which I completely agree. So the endocannabinoid system is really complicated, but it has two main branches if you think about it in simplistic ways. So one is the anandamide branch, and the other one is another molecule called 2-AG, trarachinoyl glycerol, a mouthful, but it's short 2-AG, and the two compounds are partly overlapping and partly separated, and they have different systems of production and different systems of destruction. So not only one, I think, can do phi inhibitors and enhance, potentiate what anandamide does, but one can block the enzyme that destroys 2-AG, which is called MGL, and one has other effects. And there are folks who have developed, including ourselves, who have developed inhibitors for this enzyme. And one of them is now in clinical trials. It has been now in clinical trials for about a couple of years for a variety of things: pain, epilepsy, and Tourette syndrome of all cases. And didn't turn out to be effective in Tourette syndrome, although initially there were some signs. But I'm hopeful they will be effective. So I think the endocannabinoid system, just like the opioid system, can be seen as a treasure trove of potential therapeutics. And we just need to invest, in the time and money needed to understand the system and exploit its therapeutic potential at the fullest.
That was a great way to round out the conversation. And before we finish, Dr. Piomelli, I'm wondering if you have any other closing statements regarding the health effects of cannabis and cannabinoids, any take-home points regarding the promise of cannabis or primary health concerns that the audience should keep in mind. One thing that we didn't get to, but we received a lot of questions about, is the association between psychosis and schizophrenia that we hear a lot about and cannabis use. And so that's definitely on people's minds. We didn't really have a chance to touch on it, but maybe, you can think about that in your closing comments.
Yeah, thank you for asking that. So my closing comment is, if you're talking about public health, is if you want to use cannabis, try and start after you turn 21. That is my heartfelt recommendation. I'm not saying that, I'm not gonna show you a pan with a fried egg and telling you this is your brain on cannabis, because this is not true. But what cannabis does to the brain of an adolescent, of a teenager, is much more subtle, but it is something that can last for a long time. And one of the things that it does is exactly to make you more prone to develop psychotic conditions or psychosis, like schizophrenia being one of a disease that is characterized by psychosis. Again, it's not cannabis per se that produces this directly, but cannabis interacting with the growing brain of a teenager. I know no teenager likes to hear that, but the brain of a teenager is not complete, is not fully established. We actually, for some mysterious reason, which I found fascinating, we don't stop growing our frontal cortex and our prefrontal cortex in particular until we are 23, 24, maybe even 25. So this is mysterious, because why the heck, do we have this sort of delayed development? But the fact then of the matter is that if you now, there is good evidence, I think, mostly from animal studies so far, but also from a bunch of human studies, suggesting that intervening in the interaction of cannabis with the endocannabinoid system during adolescence is problematic, because it causes subtle but very long-lasting changes. So please, if you're a teenager, if you want to try once, try twice if you like, but just don't make it a habit. Wait until you're adult, and then you can enjoy it more, just like you can enjoy more a glass of wine and stuff like that. The other situation where you probably don't want to use cannabis too much is if you're a pregnant woman, because really, literally, we don't have any idea what it does to the human fetus. We don't. It's not. I'm not saying that we know that it causes all sort of nasty things. We don't know it, and I would err on the side of caution.
Well, this was a sorry, Dr.
No, I would agree with both of those recommendations very wholeheartedly. I mean, there is a genetic susceptibility to developing schizophrenia as well, right? With cannabis, there's a couple of enzymes involved in the dopamine pathway which seem to make people particularly susceptible to this. Some genetic susceptibilities. So I would definitely stay away from cannabis. That's what I told my daughter anyway. So
I'll tell you,
she's 23.
I discovered that my daughter was smoking cigarettes. So my daughter she isn't anymore, but she was an outstandingly talented singer, opera singer when she was young. And now she still sings, but she needs the music business. She's smart. She wants to make real money. But when I discovered that she was smoking tobacco, I said, why don't you smoke pot instead? It's like, and seriously, because, to me, one cigarette of pot, one joint a day is less dangerous than 20 tobacco cigarettes in a day. 100%. And it's a question of harm reduction. Yeah. I don't like either of them, but if they have to do it, let's do the thing that harms them less. Yes. I know that I'm going to have trouble for what I just said, but it's the truth. That's what I told my daughter.
Well, this is a great way to end. And Dr. Piomelli, we didn't even get into the work that you're doing at University of California, Irvine, which is looking at the impact of cannabinoids across the lifespan. So we're going to have you come back, especially because we have about 50 questions that we haven't been able to answer. So please, this was so wonderful. And Dr. Evans, thank you so much for joining and teaching us about the opioid system as well. I want to take the time and thank both of you, as well as each and every one of you joining us today for your insightful questions. I'm sorry that we couldn't get to all of them. And of course, to the Semel staff and audiovisual team for helping with the technology, and the UCLA Cannabis Research Initiative staff for organizing this event. And as you know, I mentioned before, this programming is free and open to the community, which we do intentionally. We learned so much from you, from your questions. We do rely heavily on philanthropic support to conduct our cutting edge research and educational programs. And our goal is for you to find our webinars educational and insightful, and that they provide important information to the community about the potential therapeutic as well as adverse effects of cannabis and cannabinoids. Please find a link in the chat box for your consideration to support the good work we do at the UCLA Cannabis Research Initiative. And we'll be sending you a follow-up email with a link to the recording of this event, of this conversation. We please invite you to share this with others, and we will alert you the next time Dr. Piomelli and Dr. Evans joins us for a conversation, because there's so much activity in the chat box. We really look forward to seeing you at our next event planned for the fall of 2021. And until then, have a wonderful summer. Thank you so much.